firstly, if you just go and give me a bit of a background about what you're doing, what you've been doing for the last couple of years, or number of years in the methane space.
So what I've been doing over the last, I would say now, 10, 15 years is really looking at the microbiome, so the microbiome community in ruminant animals. And I got really interested in that aspect because they are really great systems to study to get an understanding of how different microbes work together to break down recalcitrant, so resistant biomass, plant material. And so we have been looking into the different microbes that participate in this overall process of converting that plant material into smaller molecules that then can be utilized by the animal. And when I started out, I was specifically interested in microbes that were responsible for these biomass degradation processes. Now, over the years, I shifted a little bit of my interest into microbes that are really participating in the production of methane. So while there are a lot of molecules that are being generated during that process that can be utilized by the animal, there's also a byproduct, which is methane, that is being released in the atmosphere. Now, the problem with methane when it's being released from that process is that, first of all, you lose a lot of energy. That is problematic for the animal, but in the bigger picture for people who are actually raising those animals, because you have to feed them. And so the more energy you lose in a form of methane, the more feed you have to provide. That means it becomes more costly to raise that animal. On the other aspect, what's really interesting is, unfortunately, that methane that's being released is also a huge problem ecologically. So the methane is basically functioning as a greenhouse gas. So I'm interested in not only understanding what's happening, how these microbes convert plant material partially into methane, but I'm also interested in finding solutions to how can we actually reduce methane production.
Do you want to just give a bit of background in terms of, how complex is that methane production? How many processes are there in creating methane? And I guess that's with a view of, if there's more than one way spot that it's created, or a more complex process that's created, does that also mean there's more opportunities to intervene in that process to prevent it from being created?
That's a good question. So I think that's, unfortunately, that's more complicated than I wish it would be, but that's the reason why there are so many people working on it. So I give you a little bit of maybe an idea how complicated it is and why it's so complicated. If you think about how what I'm going to say, the microbiome, which means the microbiome community, the different microbes, then we basically put it all together, we call that microbiome. And so that evolved over thousands and thousands of years. And if you think about that, we can take a cow and we can basically we provide them with different plant material. They have different capabilities of breaking that down. So they can shift their microbiome towards degrading different diets that we feed them. And so what makes it so complicated is that if you have a metabolic process that works for one particular diet, you shift the diet or you change the diet, you might also see a shift in these metabolic processes, how they really how important they are for a specific breakdown for a particular material. So it is really the ability of the animal to basically adjust to these different diets is really reflected by the different microbes that might be adjusting to these to the composition of that feed. And you also shift the different importances of these, individual processes that are taking place. So even if you develop a solution for, in one case, let's assume you're just feeding a pure corn diet, which is never happening, but just let's assume for the sake of simplicity, and then you shift that to a different feed, you will have just a totally different metabolic process that actually occurs in the rumen. And so one solution for that might work for the corn diet might not work for a different diet.
In terms of what you're looking at and who you're speaking with, what do you see as the exciting work in this space?
So I think what is really exciting, and what people have been, so the thing is that people have been trying to figure that out for the last, almost like 50, 60 years now. So it's not anything that people haven't really thought about it. The really exciting portion that we are involved in right now is that we have these techniques where we can look at microbes that we can cultivate. So in the past, people have been really struggling, or have to basically take their rumen samples and try to get these microbes in culture. So they have to grow in a laboratory in order to study them. Now, these days, there are these techniques where you can actually extract different molecules from your sample. So we can extract DNA or RNA or proteins. And then we can actually start trying to understand what these microbes do based on these molecules that we can determine. So it's like reading different books, books that we were not able to access before. What microbes are actually present and what is it that they might do. And so that's the exciting part, is really these new technologies where we can now look into microbes, what they are doing, what they potentially might do. And then we actually have these technologies where we even can go in further, where we can look at genes. So we can generate genomes, right? So we can, that's basically like the blueprint. So that would be your book. But then we can look at RNA or proteins, and that's more like, that would be in that book, let's say it's a cookbook with 100 recipes. But when you do when you look at gene expression, you basically would just read that one particular recipe. That if you go back to methane production, you look at all these different recipes that would be useful to break down all these different plant materials. But you are only interested what's happening today when they're digesting this particular feed, and then you can look specifically into those genes, into that recipe that's needed for breaking down that particular plant. And then you can even go further in. If you metabolic process for methane production, you can zoom in and you can look into that. And so that's kind of one of the most exciting parts or technologies that we have, and that become accessible to us. And we are getting to that point where we also can read, or where we can get an understanding about how different microbial populations are contributing to that aspect. So in the past, people have been mostly working with bacteria, and then also with other microbes that are called archaea. Those are really relevant for methane production. But the other aspect is there are also microbes, so fungi. There are a lot of fungi in the rumen, and people are starting to understand now that they're actually, so we didn't know until the 70s that there are actually fungi that can grow in the absence of oxygen. So it's only over the last 40 years, basically, that we understand that actually there are fungi in the rumen. And we also know by now, which has been actually really discovered by these DNA techniques, that these fungi are really important for biomass degradation. And putting it all back into the big picture is you have to degrade your biomass first. You have to generate smaller molecules that can be utilized by these methanogens, by these archaea. And so if you don't break down your biomass, or specifically, let's say fungi, then you can't take those smaller molecules and convert them into methane. So it's only over the last couple of years that we are putting together the more complete picture of the different aspects that really need to go into that recipe in order to make that work. So.
And that's awesome. That's I actually wasn't aware there was that many fungi in the rumen. So thank you for that.
So we don't know. We know the thing is that we know that there are quite a few, but we don't necessarily know how many yet. So that it's relatively unexplored, and it's relatively challenging. We understand we can cultivate them more and more. And it was only until recently that we were thinking only that there are like 4 or 5 different groups of these anaerobic fungi. And that has been expanded over the last, now I guess, 4 years, that we actually think that we have about 8 different phylogenetic groups. So it's a rapidly expanding field.
And in terms of, I guess, in terms of those dynamics, is it, this is probably a bit too black and white of a question, but how much of it, or the relationship between what's the interaction between all these species and how they're communicating, interacting, compared with just their presence? Is that something that's significant, or am I barking up the wrong tree there?
No, I think the interactions are actually the most important. So the interactions and then really the functionality. So the presence itself is important, but in the end, it's more important which of these are active. So what we see when we, if you look at time courses, you have different organisms playing a different role at different times. So when you do these studies where you look at DNA or proteins or whatever, you have snapshots in time, in time and in space, and you see differences in space, but also in time. So an example I can give you, for example, when we looked at one particular sample where we looked at the proteins being expressed, we could see that in that particular case, the fungi were contributing to some specific enzymes that are responsible for biomass degradation. But other enzymes from that have a similar function, but came from bacteria, were not important at that time. But we know that these enzymes are involved and are also produced by these bacteria. But our assumption is basically that at this particular point, the bacteria were not necessarily participating in that particular reaction. So depending on where you are looking, on the plant material in the rumen and at what time, you see a shift of these populations and what they contribute. Think about it. So these fungi, many of those are filamentous, so they grow like long filaments, and they're very slow in growing, but they're also really capable of penetrating the biomass really deeply. So assumption is that the bacteria try to colonize relatively quickly, break down some of the really easy accessible sugars.
Yeah.
Whereas now the fungi, they start to grow slowly, and they kind of almost like penetrate the plant material slowly, and they degrade material that is not easily accessible by the first batch of bacteria that basically colonize. And so there's this interplay. So you have one people come in, and so it's like, an army of people when they basically just try to, if you think about how continents were colonized. So Yeah,
Is there one that, even if it's just a gut feel, is there an area that kind of you just think, wow, that's where I'd love to throw some energy?
So I think what I believe the most promising field would be to take material that's already generated. So let's see, food waste or anything like that, that we can basically utilize and add as a feed additive or something like that. So really, and either direct feeding or extracting chemicals from small molecules from the food waste. That's where I would love to see the area going, because that would basically solve two problems at the same time. We would be able to upgrade upgrade food waste or waste in general, or at the same time utilize that. So we just reduce the waste footprint we have in that area. And then if you can basically utilize it to improve protein production from animals, I think it would be just a win-win situation, without having to think about generating something absolutely novel that would basically be a different stream. So really just utilizing what we already have and utilize that to basically solve another problem. So that's where I believe I would love to see the area going, and that would be I think the most valuable solution.
But do you see there being the same solution for grazing animals as well as fed animals, or do you think there's going to need to be two different approaches for those two different production systems?
I think they will be different. They will be different. And I think it's just like, if you think about animals that you have in a feedlot, you can just basically add some feed additives, right? So you can't have that for grazing animals, but there you might have to have a solution, either vaccine based, or it's you put something in drinking water, which is one of the sources that you can provide. So there will be different solutions, I would assume. And I think even within the same animal system, or grazing or feedlot system, there will be different solutions depending where you are. So again, it will be it's a complex problem, and there will be more solutions depending on where you are, how big your operation is. Do you have 10 animals? Do you have 20 animals? And I think, or hundreds or thousands. And I think the other thing that I believe we need to think about is really not only methane reduction, but then also really taking that and converting that into better protein.
Do you see crossover between potential solutions?
So you were breaking up a little bit, but I think what you're asking is basically just if there's the potential of reducing methane and also increasing productivity. Is that right?
Feed energy is lost as methane.
Yeah.
If that can be captured and kept within the animal, then that's the win-win.
No, I think absolutely. I think that is where it needs to go. I think that because that's kind of, unfortunately, that's kind of how the world works. It's like, unless there is some direct impact, direct benefit on the productivity side, it will not be feasible to put it into application. So I think by just reducing methane, that will not be sufficient to make a potential solution really make it to market. So it has to be both. And I think it's not an unreasonable expectation that this will be happening, because if you inhibit methanogenesis, the energy needs to go somewhere, and then so it's not. Yeah, I don't think it's necessarily an overly. If we can reduce methane production reliably, most likely what we'll see is that protein in production will increase, so efficiency will increase.
I got two more questions for you. One is, if you had the open checkbook and access to whatever equipment and whatever team you wanted to throw at this, what would you look at doing?
I think I would just expand on what we are doing right now and just trying to figure really out what is actually happening on the molecular basis or on the molecular level in the ruminant ecosystem. So really trying to figure out what are the molecules that are basically being transitioned between the different between the different microbes, and how does this change if you add different feed additives? So for example, using seaweed, and just figure out how does this really kind of affect the molecules being shuffled around in your animals, and so between the microbes, and how that all affects each other. So I think I would be really intrigued about learning about this. I think that's just the nerd in me coming out, where I just want to know what's going on there. And
then finally, is there anything interesting, exciting you've got over the next 6 months? Plans, trials, maybe just getting out of the home and traveling. I don't know. Anything interesting coming up in the not too distant future that you can share with us?
Oh, what did I share? At this point, the only thing is, right now I'm just teaching a big class, and so I'm just thinking about week to week and get to the next quarter, so that's over, and then we can focus more on research again.